about structures here for cells is that if you're going to describe structure, you got to think about the protein nature of things. So as you all know already from your freshman biology, there are 4 organic macromolecules and water and many inorganic ions that are used, that are part of the nature of things inside and outside cells in our body. Okay, but in anatomy, whenever we're going to describe the structure of how things look, the way the cell looks, the inside of the cell, the outside of the cell, how we identify a particular kind of tissue, we think about that in terms, almost always, in terms of the proteins that are there. So the carbohydrates and the lipids and the proteins and the nucleic acids that are used as organic macromolecules, carbon-based life forms, the one that we use the most to talk about structure is protein. So that's the point of this. And so if I get you thinking along those lines today, I have been successful. So this is a mock cell right here. And I want to just point out to you some places where I'm going to mention proteins for you. So the first thing I want you to notice here is that in the nucleus of the cell, we find the genetic material. Now, whenever you talk about genetic material in biology, the next thing you always say is the genes are used to give information to the cell for making proteins. That's where that goes. So at the end of this today, the very end of my protein anatomy discussion with you, we're going to learn just the sketchy outline of this. I know you did it in high school and freshman biology, but we're going to do it one more time just to point out to you the importance of protein. Okay, the next thing you notice is there's all kinds of blue dots and red dots and stuff all over in here. And if I start describing to you the ribosomes here that are in association with the endoplasmic reticulum, I'd have to talk about the proteins that are used along with the RNA to make those. It would be a protein story. If I was going to talk to you, and I'm going to do a little bit more of this today, if I was going to talk to you about cells having a mechanism within them to go through cell division, y'all learned about mitosis, meiosis, then we talk about the microtubules and the microfilaments, the networks that help divide the cells and do cytokinesis and all that cool stuff. Those are proteins. If I was going to talk about the outside of the cell membrane and its structure and the complexity of it, as I'm going to do today, I would talk about the proteins that are there. The standard model for the cell membrane is called the fluid mosaic model that you learned when you were in elementary school. What exactly is fluid about this mosaic? Proteins. If I were going to talk about the outside of the cell, things that stick out on the cell, things that help the cell move, become motor, or things that move things around the cell, I would talk about cilia or other cellular extensions off of the cell. And if I looked at the anatomy of those, I would discover they're proteins. If I were to talk about the very outside coating of most of the cells in your body, all along the inside of the blood vessel walls, epithelial cells everywhere, have this glycocalyx coating around them. And it is comprised a lot of lipid. There's a glycolipid layer there, I understand, for cell recognition. But as we begin our discussions about how cells do cell recognition, like this glycocalyx is supposed to keep the platelets from sticking. Okay, well, what about the platelets is actually causing it to stick to the outside wall of the cell? And the answer is, these are cell adhesion molecules, and guess what they are? Proteins. So wherever we go, we're going to run into this. All right, so let me introduce some of this to you to get you thinking along those lines. This is a picture we're going to look at later. This is dense, regular connective tissue. This is what tendons and ligaments are made of. And when I describe the anatomy of this tissue, I'm going to call it dense, regular. And everybody can look at and see the regular pattern here. Everybody see the nice regular stripes there? What exactly is regular? That's collagen. And guess what collagen is? A protein. Are y'all starting to get this? So I describe the anatomy of this connective tissue based on the arrangement of the protein that makes up the tissue. Here's another one. This is hyaline cartilage, and this is elastic cartilage. We're going to talk about both of these as we get to them. Now, you can look at this. You don't know anything from me yet about these types of cartilage, but you can look at this one and this one, and you can clearly see there is an anatomical difference between the two. Yes? There are cells here, and there are cells here. The matrix between the cells here looks almost glassy, this beautiful purple color. All squiggly here. We call this hyaline. We call this elastic. And why? Why are we able to tell the difference in the anatomy of these two? Well, there's something in the matrix that's different. And why would I call this elastic? As the answer you're going to learn in much more beautiful detail is there's a protein here called elastin. Those little squiggly lines actually have a unique nature to them, these proteins, elastin proteins, almost like rubber bands. And so we describe, once again, the tissue based on the cartilage, based on the protein that's in the matrix of the tissue. This is muscle. This is skeletal muscle. So I'm just giving you a smattering here so you can get a real sense of this. This is skeletal muscle, and we're going to describe skeletal, cardiac, and smooth muscle to you. We're going to look at it, talk about the differences in the two, how you tell them apart. Clearly, this has a very regular striped pattern arrangement here. Do y'all want to guess what causes that? Protein. This is actin and myosin that creates, actin is a protein, myosin is a protein, and these proteins create this striate appearance of this muscle. That's what helps us describe its anatomy. And what's more, ultimately, it's going to also help us describe its physiology. You want to describe how muscle contracts? You got to talk about how myosin and actin interact with each other. Proteins. Okay, back to the cell now. On the inside here, you see how I flipped forward just one slide there? Some of you guys are going to teach one day. It's a little clue right there. Remember your sequence, yeah? Accidentally go to the next one. See, the students all think you went there by accident. That was no accident. I did that on purpose because I'm figuring out what I'm going to say next. All right, so here's the cell again, and you can see here microfilaments, microtubules, intermediate filaments pointed to there as part of the cell. Now, what I want to say at this point is, not only do we look at the broad matrix of tissues in the inside of cells for proteins to tell what they are, but you can also back off of an individual cell. We know that we have, about 300 trillion cells in the human body, but they're differentiated. And I can look at cell types from all different places in your body and tell what they are, right? For example, I can easily tell the difference between a nerve cell and a muscle cell, and a muscle cell and a red blood cell, and a red blood cell and the egg cell, or an ovum, a reproductive cell. I can look at these under a microscope and I can tell that they're different. How is that I can tell? Well, they have different shapes, right? They have a different anatomy when I look at them. What causes that? And the truth is, you started skeleton yesterday for the human body, but the cells in our body actually have skeletons that give them their particular shape and structure. And those structural pieces are actually made out of protein. The cytoskeleton inside cells is a protein network, mostly tubulin. A couple of different varieties of tubulin used to make that cytoskeletal network. All right? Now, if I look on the outside of the cell, so I can tell the cells apart, sure enough. If I look on the outside of the cell, I can also see, so this is a single-celled paramecium. I just wanted to show you, this is true for comparing different life phenomenon. Every freshman textbook has a slide somewhere like this in the first chapter where it says all life is related. And the textbook points to the fact that these single-cell organisms use cilia, cellular extensions made of protein, and very sophisticated epithelial linings within the respiratory system use cilia, not in this case to move themselves, but in this case to move product So this is a pseudostratified columnar epithelial section, and goblet cells here produce mucus that end up on the outsides of the cells, and that creates a river of mucus within my body that moves like a river does. And the movement of that river is due to the organized contraction of cilia on the outsides of these cells. And those cilia are made of protein. In fact, if you take a picture of the cilia, if you cut a transverse section of cilia off of a paramecium, a prokaryotic single-celled organism, and you compare it to the cilia on the pseudostratified columnar cells in the human respiratory system, they are identical. They look the same. Now, if we break the structure down of the cilia or the flagella, flagella would be like a cilia on a sperm cell. We now know, because of the work of molecular biologists, that these things are made up of very organized arrangements of proteins. It's called a 9+2 arrangement, and you can see in this cross-section of a cilia the arrangement of these proteins and how they're held together. Now, as it turns out, the arrangement of these cilia, the structural pieces on the outside of the cell made of proteins, we now know, as I said, because of the molecular guys who've been doing this now for the last couple of decades, that it is an extremely organized system of proteins that work in ways that we can now explain. And so I've got a picture of the guy who's written a very famous book, Darwin's Black Box, and I'm sure some of you have heard of it, but he goes through the anatomy of this in layman's terms so everybody can understand it, people like me who don't have a molecular biology background. And what he points out is, in his first book, he points out that this arrangement is very organized. It has patterns. And if you do things to the arrangement of these proteins, you cause all kinds of problems for cells. You don't, if you knock out one of these guys so that they're not put together properly, the thing unravels. And so Behe uses the structure of this protein in his first major book to say it is impossible to explain these structures, the anatomy of these structures like cilia, in terms of a Darwinian model. You just don't read about Darwinian mechanisms at this level. So in most freshman textbooks, the A&P textbook doesn't discuss it at all, but most freshman textbooks will go through a picture like this and tell the students, well, look, this observation that the cilia and the cilia are the same structure suggests that all life is related. And some